Greetings of the day. Today we will see the third week of the development. So which includes these are the main stages of the which happens in the third week of the development. So first is the gastrulation, next is the primitive streak, and next is the notochord and neural tube, intra-embryonic mesoderm, somites. Of which the formation of the primitive streak, the formation of the notochord, fate of the intraembryonic mesoderm, and fate of the somites. So these are the important questions which usually we will see in the 6 mark section or the so-called short answer section. Okay, so we will see about how the different events happen in the third week of development, that is the gastrulation, primitive streak, and then the notochord, neural tube, intraembryonic mesoderm, and their fate. And the lastly, we will end this video by the learning about the somites, number of the somites, and formation of the somites. So now, as I said, the first one we will be seeing it is about the gastrulation. So, what is first of all we have to understand what is meant by gastrulation. So, the gastrulation refers to the formation of the three germ layers. So, that is called as the gastrulation. So, basically, it is defined as the formation of the three germ layers. So, that is the first one, that is the ectoderm, that is the which is also called as the outer or surface layer. Outer or surface layer. Now, next one, the second one, that is the mesoderm or the middle layer. Mesoderm or the middle layer. And lastly, the endoderm. The third one, that is the endoderm, that is called as the deepest layer. That is the deepest layer. So basically gastrulation can be defined as the formation of the three germ layers. So these are the three. Okay, in that one, after this one, let us see about the first of all, before understanding the formation of the three germ layers, let us have an idea about the primitive streak. Okay, so the primitive streak, first of all we will understand about the formation of the primitive streak, fate of the primitive streak, and functions of the primitive streak. So we will be seeing about the primitive streak. So in the bilaminar germ disc, what happens is that the first sign of the gastrulation, it is the appearance of the primitive streak. So what is actually the primitive streak? It is simply to say that is an appearance of the linear thick band on the posterior. So as it is embryology, we have to use the word dorsal. So linear thick band on the dorsal surface of the bilaminar embryonic disc at its tail end, or simply calling as the caudal end in the midline. Okay, so let us define still more simpler. Linear thick band on the dorsal surface of the embryonic disc, which is basically bilaminar, and it is forming at which end? That is the caudal end, and where exactly? That is the midline. Okay, so and now how it is formed? So mainly it is formed due to the proliferation. Mainly it is formed due to the proliferation and migration. Proliferation and migration of epiblast cells. Proliferation and migration of the epiblast cells, that too in the midline. So epiblast cells in the midline. So this is how and why the primitive streak is forming. And now the cells forming the primitive streak, the point to be understood it is that the cells forming the primitive streak, they are pluripotent. Cells forming are pluripotent. So which have got a capability of forming all type of adult cells. Forming all type of adult cells. So that is about the primitive streak, the basic definition, and it appears on the most important timeline, that is on the 15th day. that is on the 15th day of the embryonic development. 15th day of the embryonic development, we can see the formation of the primitive streak. So let us try to understand the picture on a bilaminar disc. In the previous video, we have seen the formation of the precaudal plate. So the precaudal plate, which is on the cranial end. So this is labeled as the precaudal plate. This is labeled as the precaudal plate, and here we will be seeing the formation of the cloacal membrane. So this is the cloacal membrane. Now we are seeing the formation of the primitive streak. So let me show with a different color here. So this is the primitive streak, and now yes, so this is the primitive streak. And now after appearing on the 15th day, what is happening is that the cranial end of the primitive streak, it presents a rounded elevated area called as the primitive node. Okay, so this is the rounded elevated area that is called as the primitive node, or also called as the Hansen's node. Primitive node, also called as the Hansen's node. And now the center of the primitive node shows a depression. So that is the depression, and that depression is called as the primitive pit. So that depression is called as the primitive pit. That depression is called as the primitive pit. And now what is happened after that, a narrow groove appears, and that groove appears in the midline of the primitive streak, and that is called as the primitive groove. So now from the groove exactly, yes, okay, so now this is the complete picture of the formation of the primitive streak, and now we can label this as the primitive streak. So So that is the complete formation of the primitive stick. So let us see in a simpler ways. So first of all, in the bilaminar disc, on its dorsal wall, there will be the linear thick band that is called as the primitive stick. And why it is formed? Because the proliferation of the cells, proliferation and migration of the epiblast cells. So these are the two important words to be understood: proliferation and migration of epiblast cells in the midline. So now the cells forming the primitive streak, they are the pluripotent, and what happens is that first of all, the cranial end, it presents a rounded elevated node called as the primitive node, and after that one in the center there will be a depression that is called as the primitive pit, and after that one a narrow groove appears in the midline that is called as the primitive groove, and that primitive groove is continuous cranially with that of the primitive pit. So this is how the primitive streak is formed. Now after this primitive streak is formed, the important step which is happening is that the formation of the three germ layers. So let us try to understand the formation of the three germ layers. So now let us see the formation of the three germ layers. So here what is happening is that after the primitive knot is formed or the primitive streak is formed, there what happens is that the cells of the epiblast they migrate. Okay, so actually from the epiblast, let me write here. So the cells of the epiblast migrate here. So cells of the epiblast migrate to form the three germ layers. To form three germ layers, and what happen? So what do they do? Where do they migrate? Is that they will be migrating here and move inwards towards the hypoblast. So some of these cells replace the hypoblast cells to form the endoderm. Okay. So let me show with the diagram. So first of all here. So this is the basal membrane, and here we will be seeing the cells with their respective nuclei. So with the under color, let me try to show you. This is the flattened cells. So now what is happening is that so this is the epiblast cells. This is the upper epiblast cells, and here this is the hypoblast cells. Here that is the hypoblast cells. Now here what is happening is that the cells will be migrating in this direction. Okay, so the cells will be migrating downwards and here invaginates, and now due to which we will be having the new type of cells. And here one more important implication we can see is that here the third germ layer will be seen, that is called as the mesoderm cells. So here the third color which I have been using, that is the mesoderm cells. That is the third layer. So this is the mesoderm. So this is formed due to the migration, proliferation and migration of the cells. So that is called as the that is the third layer. And here this is the and later what happens is that this further will be developing into, after this one they will be closed, so that nicely we can see the newly formed the three layers of bilaminar disc will become the trilaminar disc. So the first one as usually, that is the epiblast. Okay, so that is the first one, and next one that is the second, that the next one, that is the endoderm, which is here a part, something like that of the flattened cells. So here, and then next comes the third layer in between them, that is the mesoderm cells. So this is how the bilaminar will become the trilaminar and starts showing, it shows the three layers. So now let us label this one. So this is the ectoderm, which is formed from the epiblast. This is the mesoderm, So that is also formed from the epiblast, and here this is the endoderm, which is also formed from the epiblast. So we can conclude that these three are being formed from the epiblast cells. Okay, so this is how the three germ layers are formed, and this is called as this is basically referred as the gastrulation. Now here what is happening is that the next one we will be seeing is that, and before that one, sorry, yes, so that is the formation of the this is how the three layers are formed. And now here what is happening is that the mode of spread of the intra-embryonic mesoderm, mode of spread of the intra-embryonic mesoderm. So now here in the bilaminar disc, let us see how it is seen. So this is the bilaminar disc, and here it is the precaudal plate, and here this is the cloacal membrane, and here this is the primitive streak with the primitive node, primitive pit. And now here the migration of the cells will be following in this arrow marked ways. Okay, so one, and here second, and here that is the laterally one, two, and downwards. Okay, so this is how the cells will be migrating. Okay, so these arrow marks are the migration. These cells are the migration, and now the same level. So this is the precord procaudal plate, and here this is the cloacal membrane. So here this is the cloacal membrane. So now the cells are getting migrated in this way. So now after this one, this is how the finally primitive groove, primitive streak, and the three layers of the germ layers are formed. So first of all, so in a serial way, first that is the primitive streak will be formed, and then the migration of the cells will be formed, due to which the three germ layers are derived. And after that one, that the mesoderm will be migrating. And now what is happening is that the intraembryonic mesoderm spreads on the cranial, caudal, and lateral direction, except the region of the precaudal plate. What happens is that they don't migrate here. So they don't migrate here. And then so this is the region. So this is the region, they don't migrate. And what do they do is that their ectoderm and then the endoderm are in the firm contact, and they form the future buccopharyngeal membrane. So first one, and here the second one, that is the cloacal membrane. There also the ectoderm and the endoderm, they are close contact with each other. And the third one, that is the notochord. So these are the three regions. So one, second, and the third. So these are the three regions where what is happening is that the intraembryonic mesoderm will not be seen except in these three following regions. Okay, so now with this what we have learned till now is that the formation of the primitive streak, formation of the three germ layers, and that is the intraembryonic mesoderm and their exemption. So before going to the most important topic for today, that is the notochord, let us try to understand the functions and the fate of the primitive streak. So first of all, that is the functions of the primitive streak. So now here first of all the functions of the primitive streak already in the previous in the initial part of the video we have seen that the ectoderm and endoderm is formed first and then primitive streak is formed and then it is giving rise to the third germ layer, that is the intra embryonic mesoderm. So that is the intra embryonic mesoderm. So that is the first function of the primitive streak. And later what does it do is that formation of the septum transversum. So in the previous picture what we have seen is that the migration of the mesoderm will not be cranial to that of the procaudal plate, and due to which there exactly the septum transversum is formed. So cranial to the precaudal plate, what is happening is that there will not be the there will not be the migration, and there the septum transversum is formed. And then the third function, that is the most importantly, that is the formation of the notochord. Formation of the notochord. Formation of the notochord. And lastly the it determines the craniocaudal axis. Craniocaudal axis of the embryo. Craniocaudal axis of the embryo. And then most importantly demarcation of the right and left halves. Demarcation of right and left halves. Demarcation of the right and the left halves. Okay, so these are the functions of the primitive streak. And now what is happening, so these are the basic functions. Now after this one let us try to understand the fate of the primitive streak. Fate of the primitive streak. Formation is done, functions is done. Now what happens to the primitive streak? Now here it actively forms the intraembryonic mesoderm up to the third week of the intrauterine life. Third week of intrauterine life. And now after this one it regresses. So after this one the regression happens craniocaudally. So from the cranial end to the caudal end, and completely disappears by the fourth week. So fourth week by fourth week. So we can write that by fourth week the primitive streak will be disappearing. Now at this juncture let us understand, is there any clinical correlation? Yes, actually the clinical correlation is that the primitive streak undergoes the degeneration. That's what we have learned just a few moments back, and that should end by the fourth week. But unfortunately what happens is that in some conditions the remnants of the primitive streak will be persisting, and they give rise to a large precoccygeal tumor. The large sized precoccygeal tumor, that is called as the sacrococcygeal teratoma. Sacrococcygeal teratoma. So which forms sacrococcygeal teratoma, which happens and which forms due to the persistence of the remnants of the primitive streak even after the fourth week of intrauterine development, and its large size may cause an obstructed labor or even death of the baby, and its occurrence is 1 is to 37,000 births. Okay, so 1 is to 37,000 pregnancies, and it occurs more in the female babies. And what is happening is that it may become malignant, usually becomes malignant during infancy, and that is why before the sixth week only, sixth month it should be removed. And as it is located at the coccygeal region, that is why it is called as the sacrococcygeal. And excessive blood flow also may cause the heart failure, which is also called as the hydrox. So these are the clinical correlations of the primitive streak. So first the formation, that is the between the precaudal plate, and then after that one the cloacal membrane. In between them in the midline there will be the migration and proliferation of the cells, and that is called as the primitive node, primitive pit. Okay, and from there the proliferation and migration of the cells happen in all the directions except at two, where that is the precaudal plate and also the precaudal plate and also the cloacal membrane, where the ectoderm and endoderm they will be intact. And now after this one This is how the primitive streak is formed. The functions are seen here: intra-embryonic mesoderm formation, septum transversum formation, notochord formation, craniopodal axis of the embryo presence, and then demarcation into the right and left halves. And now comes the most important question and most important concept in the general embryology, that is the formation of the notochord. Okay, so with the formation of the notochord, the very important event happening in this third week can be understood, and that can be understood very easily by a series of diagrams. So stages of the development. So now let me write here, that is the stages of development of notochord. Stages of development of the notochord. So which can be understood by a series of diagrams. So here what is happening is that before starting, that is the let us define what is the notochord. So notochord can be defined as the midline cord of cells that develops in the region. So midline cord of the cells. Midline cord of the cells. So that develops in the region between the primitive streak, between the primitive streak and the precaudal plate. Primitive streak and precaudal plate of the embryo. Precaudal plate of the embryonic disc. So that the and it develops from the primitive knot, that is also called as the Hensen's knot of the primitive streak. So now first of all, what happens is that the cells of the primitive knot of the primitive streak, they proliferate to form the prenotochordal cells, so that moves the inward and to produce a central depression called as the blastopore. So now let me explain with the diagrams, that is a series of the diagrams as I have already mentioned. So after understanding the definition, and now we will be directly seeing the formation of the notochord in a series of the diagrams. So first of all here, so this is the upper layer, and here now let us label water. First of all, here this is the cloacal membrane, that is the cloacal membrane. And here what is happening is that the proliferate to form the prenotochordal cells, and that moves inwards to produce the pit. So this is the area. What is happening is that for proliferation inward, to move the inwards, they will be directing themselves inside, due to which there is a depression forming, that is called as the blastopore. That is called as the blastopore. So let me repeat once again. The cells of the primitive node will be developing, will be proliferating, and then they will be invaginating. So there is a site of invagination, that is called as the blastopore. Now what is happening is that from the bottom of the blastopore, what happens is that the cells will be migrating, and where exactly? In the midline and between the ectoderm and the endoderm, to form the solid cord of the cells called as the notochordal process or the head process. Now what is happening is that, now let me show with the color difference. So this I have mentioned that this is the form blastopore, and now the cells are proliferated and they started migrating. So this is the thick cord of the cells, and now let me label, this is the formation of the notochordal process. Notochordal process, and here these combinedly we can name them as the precaudal plate. Precaudal plate. So, in the first step, what is happening is that the notochordal process is being formed. Where exactly? On the surface ectoderm, where the primitive knot is there, they will start and they form a central depression. That depression is called as the blastopore. Now, what is happening after this one is that the notochord increases in the length. And after this one, by extending caudally, and as the primitive streak recedes caudally and regresses. So now, due to which what is happening is that here I have drawn a solid cord-like structure. Now in the next picture, what we will be seeing is that they become a canal-like structure that is called as the notochordal canal. That is called as the, so now here the solid cord-like cells, what it will become is that in the next level, in the next stage, they become a canal-like structure. They became a canal. So here is the first one, first layer. This, the precaudal plate. Now here, what is happening is that the, now I will be drawing only the canal. It is no longer the cord-like structure. So it is just a canal. It is just the canal. So now I will label this as the notochordal canal. Now I will label this as the notochordal canal, and the labeling remains the same. So this is the precaudal plate, and here there will be the cloacal membrane. Cloacal membrane. Okay. Now, after this one, now the most importantly here you can see the fusion. What happens is that the fusion of the notochordal canal, the fusion of the notochordal canal with the endoderm happens. So, same once again. So, let me show with the diagram. First layer, and here it is the second layer. So what is happening is that the fusion of the with the floor. Okay, so here you can write that is as a fusion of the floor. Fusion of the floor of notochordal canal with the endoderm. So that is the third step. So here this is the first one, second one, and here this is the third one. Now here after this one, in the next layer, what happens is that in the fourth step, after the fusion, there will be the breakdown of the later both the fused parts, they will be breakdown, and due to which we have to remember still it is a in a sac like structure. So that is why it communicates above with the amniotic and below with that of the yolk sac. So now in the fourth stage, so remember this is the fourth stage. So let me try to draw the diagram here once again, the same. So the two, and here this is the procaudal plate. So now so the breakdown has happened, and here I am drawing it. Okay, so now here this is the breakdown. So here I will write that it is the breakdown of the fused floor. Breakdown of the fused floor. Now that is the fourth stage. And now after this one, what is happening is that the gradually the walls of the canal become flattened to form a plate-like structure called as the notochordal plate. So now after this one, once again the next one, that is the stage 5 of the formation of the notochord. So here I will write that is the fifth stage. So what is happening is that the formation of the plate-like structure, that is the notochordal plate. And so this plate will be forming like this. Okay, so this is how it is closed. And here this is now this can be labeled as the notochordal plate. Now after this one, what happens is that in the next, that is the sixth stage, the notochordal plate gets curved to form a canal. And after this one, a definitive, the proliferation of the cells of the tube will be forming the canalization and converts into a solid. After that canal has formed, and then there will be solid cord of cells called as the definitive notochord. So that is in the last stage. So let me show with a diagram. So here what is happening is that the sixth stage you can number it as. And now, so the first top layer, okay, and after this one the bottom layer. So here I am drawing a solid cord of the cells. Yes. So this is the definitive notochord. This is the definitive notochord. Please remember, this is a very important concept and also very important question always in the general embryology, that is the definitive notochord formation. So here what is happening is that, first of all, let us revise here. The first stage, the cells of the primitive knot of the primitive streak, what do they do is that they proliferate to form the prenotochordal cells, and that moves inwards from a central depression, that is called as the blastopore, we have seen. And from the blastopore they will be migrating as the solid cord of cells, that is called as the notochordal process, that we have seen. And after this one, the notochordal process, what happens is that they canalize to form the notochordal canal. So that notochordal canal, what does it do in the next level? That is fuses with the floor. And after it fuses, what happens is that there will be a breakdown, due to which there is a communication. So here one, and here that is the second breakdown of the fused floor. And after that one, the formation of the notochordal plate. And lastly, there will be they forms a solid cord of cells called as the definitive notochord. So now, due to which what is happening is that, here if you can understand, the endoderm is restored, and now the notochord is completely separated. So you can see notochord is completely separated from that of the endoderm. So that is about the notochord formation. And now let us try to understand the functions. Okay, so the functions of the notochord. There are very important functions here as such for the formation of the notochord. Okay, so the central axis, formation of the central axis, and then formation of the neural tube from the overlying ectoderm. So that is another important concept that we will learn after this one. and after that one, that is the central column around which the vertebral bodies and intervertebral discs will develop. Central column. Okay, so after this one, now the fate. Okay, so the fate is that most importantly what do they do is that they form the, they actually they all they are present in all the animals belonging to the phylum chordata. And after that one, the notochord appears only in the embryo. In later life it disappears, and most importantly they remnants are seen in the form of nucleus pulposus. Okay, so very important MCQ here. So what is the remnant of the notochord? So let me write it. So that is the nucleus pulposus. Nucleus pulposus. So of the intervertebral disc and also the apical ligament of the dens. Now the clinical correlation as usually. So there is a condition called as the chordoma. There is a condition. What is this? This is a rare tumor that arises from the remnants of the notochord, and it is seen either in the cranial or in the sacral region, and usually it is seen in the males, that too late in the life over 50 years, and about 30% of these can turn into they are the malignant, and they are seen at the base of the cranium and has a tendency to even spread into the nasopharynx. And now once again let us see the formation of the neural tube, which is an another important concept, which is followed by the intra-embryonic mesoderm. So now in this at this point we will see the formation of the neural tube, which is defined as the neurulation, which is defined as the neurulation. So the formation of the neural tube is called as the neurulation. Here also we require a series of diagrams so that we can understand the formation of the neural tube. Okay, so now the source is the ectoderm. So the neural tube is formed from the ectoderm. So the source has to be remembered as it is an important MCQ. The neural tube, it is the notochord induces the overlying ectoderm. Okay, so to differentiate into special cells called as the neuroectodermal cells. So now let us draw a diagram here. So that is the first layer as usually. So now we will label this as the endoderm. And next one here, that is the ectoderm. So we will show that color differentiation here. And now here the proliferate, they will be proliferating to form a thick band. To form a thick band. So now I will label this as the neural plate. Now I will label this as the neural plate, and here this is, as I said already, this is the ectoderm, and in the central line we have seen already the formation of the notochord. So endoderm is there, ectoderm is there, and here this is the neural plate, which is a thick proliferation. So that is called as the neural plate. And now where does it extends? In the midline from the primitive knot, sorry, from the precaudal plate to the primitive knot. So let us see its extent. So that is the precaudal plate, precaudal plate to the primitive knot, precaudal plate to the primitive knot. So this is how the extent is seen. And now what is happening is that the the margins of the neural plate gets elevated. Why? Because most importantly deep to that one we can see the paraxial mesoderm proliferation. Okay, so due to which there forms the folds. Okay, so the neural groove becomes deeper and the neural folds this leads to the formation of the neural groove on bounded on either side by the neural folds. So let me explain with that of the diagram. So what we have seen is that first of all the proliferation has done, and now the neural groove will be formed. So this is how the neural groove is formed, and here the same older one. So this is the notochord, and here this is labeled as the neural groove. Okay, so due to the proliferation of the mesoderm, that is the paraxial mesoderm, these regions have got elevated, and due to which they formed the grooves. Okay, so and now these elevated parts we will name them as the neural fold. So I will label this as the neural fold. So now after this level, after this form, what happens is that the groove becomes deeper. Okay, so the groove becomes deeper, and the neural folds form fuse to sorry, they become deeper and move towards midline to fuse each other and to form a cylindrical neural tube. Now most importantly, please try to listen carefully and understand properly. The fuse to form the cylindrical neural tube, and the fusion of the neural folds begins in the middle and extends cranially, extends in the cranial and caudal direction. So before the complete torsion of the fusion, the neural tube communicates with the amniotic sac and with the two openings. One is the cranial and another is the caudal. So they are respectively called as the cranial neuropore and caudal neuropore. So now let me draw the diagram here first of all. So the above one, what I have said is that this is the neural folds have developed like this, and they started developing here. They started developing, and after this one, so this is the straight line here, and now yes, with a different color. Okay, so now these tips are called as the, where I am drawing in a dark line, that is called as the neural crest. So this is called as the ends which are uh, solid cord like structure, that are called as the neural crest. And here as usually the notochord is seen, and what happens is that the fusion of the neural folds happens in the next level. So let me draw here. So in the last stage, the baseline here, and then so here it is formed. And now let me draw, this is the, here in the central line, that is the neural tube. This is the neural tube, and here is the notochord as usually, notochord as usually. And now here you can see the new masses of the cells, new masses of the cells, that are called as the neural crest. So altogether we have got neural, so let me label, this is the neural crest, this is the neural crest, and here this is the neural tube, and here this is the notochord. So this is how the notochord is formed. Okay, so the neural tube forms the what is happening is that the neural tube will be forming the central nervous system. Okay, so the neural tube, it forms the central nervous system, and while the neural crest cells, they form the peripheral nervous system. Okay, so this is how the neurulation or the formation of the neural tube happens. So first of all, here the surface ectoderm cells, which is overlying the notochord, will be thickening to form the neural plate, and the neural plate will be showing the fold-like structure in the next picture we have seen. Why? Because the intra-embryonic mesoderm will be developing, and after that one, later they form the they will be formation of the neural groove formed by the neural folds, and then they become deeper. And what is happening is that they become deeper and neural folds move to fuse, and due to which their ends will be forming a separate group of cells called as the neural crest cells. Neural crest, they are forming the PNS or the peripheral nervous system, and the neural tube, they are forming the CNS. Okay, so now after this one, we will be seeing the formation of the intra-embryonic mesoderm and their subdivisions, and then the formation of the somatomeres, and then the somites, with which we will end the video. So now here, first of all, we will be seeing the next. We will be seeing the intra-embryonic mesoderm. So as the name itself is saying, it is in the within the embryo. So that is why we will call that as the intra-embryonic mesoderm, and it is derived from the primitive streak. So it is derived from the primitive streak. Is starting in the starting of the video we have seen, that is from the primitive streak, and it lies between the ectoderm and endoderm except at the precaudal plate, notochord, and then the cloacal membrane, and it lies on either side of the notochord. And now what is happening is that in front of the, or you can say the intraembryonic mesoderm of the two sides meet in front of the precaudal plate to form the cardiogenic plate, so that we once again we will get in the development of the CVS. Now, by the diagram, let me explain the subdivisions of the intraembryonic mesoderm. Okay. So here, the first of all, that is the precaudal plate, and then here at the end, that is the cloacal membrane, and here that is the primitive node, and here this is the primitive streak. Now what is happening is that we will see the formation of the formation of the intraembryonic the divisions. Now here first of all, let me draw with a dotted line, that is the notochord. This is the notochord. So till that we will label first of all, that is the precaudal plate, that is the precaudal plate, and here this is the primitive knot, and here this is the primitive streak, and here this is the cloacal membrane. This is the cloacal membrane. Now here we can see the formation, we shall see about the formation of the mesoderm. Okay, so let us show the color difference. Now, so exactly on either side of the notochord, we can see the formation of the, we can see the development of the paraxial mesoderm. Okay, so paraxial mesoderm, let me draw with the horizontal lines for our understanding. So this is the paraxial mesoderm, paraxial mesoderm. Paraxial mesoderm. Now after that one, extreme laterally, they will be seeing the formation of the lateral plate mesoderm. Okay, so now here with the color differentiation, so this is all the lateral plate mesoderm. So this is the lateral plate mesoderm. This is the lateral plate mesoderm. And now in between these two we can see the intermediate mesoderm. So with the color differentiation, so now I will label this as the intermediate mesoderm. This is the intermediate mesoderm. So on exactly on the side of the lateral to the notochord, that is the paraxial mesoderm, so which I have drawn with the horizontal lines, and extreme laterally with the red color what I am showing, that is the lateral plate mesoderm, and in between them that is the intermediate mesoderm. Now after this one, the paraxial mesoderm, what happens is that it let us see the fit. The paraxial mesoderm, it divide undergoes into somatomeres. So it undergoes the segmentation. So that is the somatomeres, and that is about the paraxial mesoderm. And now the intermediate, later this one, the intermediate mesoderm. What happens is that they form the kidneys, testes, and ovaries. So they form the most of the genitourinary. And the lateral plate, they form the body wall and the body cavities. And later it is also forming the intra-embryonic coelom. So in the briefly, let us see the development of the somatomeres. So somatomeres already I have said now, that is seen in the, that is derived from the paraxial mesoderm. So that is the paraxial mesoderm. So it undergoes the segmentation to form the somatomeres and then the somites. So first of all, the paraxial mesoderm, that is a, we can say that is the longitudinal thick column. Longitudinal thick column. Thick column of the mesodermal cells, and that lies on either side of the notochord and the developing neural tube. And what does it do is that it undergoes segmentation and gets organized into the mass segments called as the somatomeres, and they are the, as I have said, loose masses of the paraxial mesoderm. Okay, so the first pair, what happens is that the first pair appears in the cephalic region, that is the head region. So that appears in the cephalic region and proceeds caudally, craniocaudally. And now they are numbered, and the development is that first of all, they are the cubical blocks of the paraxial mesoderm and first appear in the future occipital region. So now the first pair, they forms on the 20th day. So they form on the 20th day, important timeline here. So subsequent forms, they appears in the craniocaudal sequence. So now you can say cranial somites are the oldest and the caudal somites are the youngest. So altogether about 38 pairs will be formed. About 38 pairs are formed between the days 20 and 30 of the development. So by the end of the fifth week, what is happening is that all the 44 pairs of the somites are formed. And now after this one, let us see about the correlation between the somites and the spinal nerve. So as discussed earlier, though they form at the cranial and they go on continuing in the caudal direction. So now we will name them as the occipital, cervical, thoracic, lumbar, which more or less correlates with the number of the spinal nerves. Okay, so now the doubt may come regarding the number of the coccygeal somites, but what is happening is that some extracoccygeal somites degenerate. And now most importantly, the structure of the each somite. Okay, so each somite, let me draw a diagram here. So it is divided into three parts. So more or less it is in the triangular. Okay, so that is the central notochord like structure. And once again, Yes. Okay. So this is how the somite is seen, and which contains the three parts. First, second, and then the third part. So the medial most one is called as the sclerotome. So the medial most is called as the sclerotome, forms the vertebrae and then the ribs. Vertebrae and ribs. And now after this one, here the central one, that is called as the myotome, which contains a vesicle. So myotome, so that is the myotome. And what do they do? That is the myotome which forms the skeletal muscle, as the name itself is suggesting. So skeletal muscles. Skeletal muscles. And lateral most, that is the dermatome. Laterally, that is the dermatome, which forms the dermis of the skin. Dermis of the skin. So parts of the somite is an important 3 marks question. So medial most, that is the sclerotome forming the vertebra and the ribs, and middle is the myotome forming the skeletal muscles, and the lateral is the dermatome which forms the dermis of the skin. And now the intermediate mesoderm, already the fate we have seen, that is the kidneys, testes and ovaries, and lateral plate, they form the body wall and then the body cavities. And also it is formed in the formation of the intraembryonic coelom. So briefly let us see the formation of the intraembryonic coelom. So this is the disc. Here it is the precaudal plate, and what happens is that the lateral plate of mesoderm is continuous with each other, and a small cavities appear, due to which there forms the coelom. So here we can see, so that is called as the intraembryonic coelom. That is called as the intraembryonic coelom. And here what is happening is that cranial to that one, here we can see a gap, that is called as the septum transversum. Septum transversum. Okay, so the it is divided into the it is the intraembryonic coelom. And now what is happening is that the part anterior to the precaudal plate, that is called as the pericardial cavity. So here this is the precaudal plate, and anterior to it, that is called as the pericardial cavity. So here pericardial cavity. So and now we will call this as the pericardio-peritoneal canal. Pericardio-peritoneal canal. So and so this is how. And now the right and left limbs, you can say this is the right limb and here it is the left limb, and they form the peritoneal cavities. So that is how the intraembryonic coelom is formed. In the intraembryonic mesoderm, the small cavities appear and later they coalesce to form the cavity. All these cavities coalesce to form a single large horseshoe-shaped cavity, that is called as the intraembryonic coelom. And now after this one, as usually, once again there will be the formation of the somatopluric and then the splanchnopluric layer of the mesoderm. So due to which what is happening is that the somatopluric lies in contact with that of the ectoderm. So in the cross section, let us see. Okay, so in the central, that is the notochord, as usually, sorry, neural tube. This is the notochord, and paraxial mesoderm with the longitudinal for our convenience. Okay, and then the intermediate mesoderm with an empty cavity, and then that is the lateral plate mesoderm with an intra with a coelom. Okay, so that is, and now the intra so this I will be labeling as the intra embryonic coelom. And now what is happening is that the somatopleuric layer, it lies in contact with that of the ectoderm. So the above one, so here, that is the somatopleuric mesoderm, which is in contact with that of the ectoderm. So this I will labeling is a somatopleuric mesoderm, and here that is the splanchnopluric mesoderm. Splanchnopluric mesoderm, so which is in contact with that of the endoderm, that is called as the splanchnopluric mesoderm. So somatopleuric here, that means here they will be contributing to the development of the body wall, whereas the splanchnopluric, they contribute to the walls of the viscera. So one is walls of the body and another is walls of the viscera. So that is the how the somatopleuric and splanchnopluric they are formed, and they are contributing to the different contents. And now here the intraembryonic mesoderm lying anterior, which is here we can see the septum transversum, and which contributes to the formation of the liver and diaphragm. Liver and diaphragm. So this is how the liver and diaphragm is formed. So now with this I will end the video. Thank you very much.